Metabolic integration and organ specialization. The liver. The liver serves as a major metabolic processing center in vertebrates, except for dietary triacylglycerols, which are metabolized principally by adipose tissue. Most of the incoming nutrients that pass through the intestinal tract are routed via the portal vein to the liver for processing and distribution. Much of the liver's activity centers around conversions involving glucose 6-phosphate. Glucose 6-phosphate can be converted to glycogen, released as blood glucose, used to generate NADPH and pentoses via pentose phosphate cycle, or catabolized to acetyl-CoA for fatty acid synthesis or for energy production via oxidative phosphorylation. Most of the liver glucose 6-phosphate arises from dietary carbohydrate, from degradation of glycogen reserves, or from muscle lactate that enters the gluconeogenic pathway. The liver plays an important regulatory role in metabolism by buffering the level of blood glucose. Liver has two enzymes for glucose phosphorylation, hexokinase and glucokinase. Unlike hexokinase, glucokinase has a low affinity for glucose. Its Km for glucose is high, on the order of 10 millimoles. When blood glucose levels are high, glucokinase activity augments hexokinase in phosphorylating glucose as an initial step leading to its storage in glycogen. The major metabolic hormones, epinephrine, glucagon, and insulin, all influence glucose metabolism in the liver to keep blood glucose levels relatively constant. The liver is a major center for fatty acid turnover. When the demand for metabolic energy is high, triacylglycerols are broken down and fatty acids are degraded in the liver to acetyl-CoA to form ketone bodies, which are exported to the heart, brain, and other tissues. If energy demands are low, fatty acids are incorporated into triacylglycerols that are carried to adipose tissue for deposition as fat. Cholesterol is also synthesized in the liver from 2 carbon units derived from acetyl-CoA. In addition to these central functions in carbohydrate and fat-based energy metabolism, the liver serves other purposes. For example, the liver can use amino acids as metabolic fuels. Amino acids are first converted to their corresponding alpha keto acids by aminotransferases. The amino group is excreted after incorporation into urea in the urea cycle. The carbon skeletons of gluconeogenic amino acids can be used for glucose synthesis, whereas those of ketogenic amino acids appear in ketone bodies. The liver is also the principal detoxification organ in the body. The endoplasmic reticulum of liver cells is rich in enzymes that convert biologically active substances such as hormones, poisons, and drugs into less harmful byproducts. Liver disease leads to serious metabolic derangements, particularly in amino acid metabolism. In cirrhosis, the liver becomes defective in converting ammonia to urea for excretion, and blood levels of ammonia rise. Ammonia is toxic to the central nervous system, and coma ensues. And that's metabolic integration and organ specialization, the liver. Have a great day.